12 years, $2.5 billion, and a staggering 90% failure rate.
Yeah, it's brutal.
Right. I mean, right now, those are the underlying economics of the $1.5 trillion global pharmaceutical industry. Bringing a single novel drug to market it's just an absolute mathematical nightmare.
It really is. I mean, it's an industry that's built entirely on a foundation of just blind stochastic guessing.
Yeah, and I want everyone to hold those numbers in their head for a second. 12 years, $2.5 billion. Now, contrast that with this. It is now 2026. An AI model can hallucinate a novel, perfectly functioning protein designed to cure a specific form of cancer, and it can do it in just 48 hours.
Yeah.
And the cost is literally just the price of your cloud compute bill.
Exactly.
So the era of biological discovery is dead. The era of biological engineering has officially begun.
Yeah, we are basically watching the greatest financial moat in modern history just evaporate in real time.
Welcome to MediaGenX. I mean, what you just heard is the sound of a massive paradigm shift. And our mission today for this deep dive is straightforward, but it's massive. We are tearing down the legacy pharma cartel, and we are mapping out what we call the open source biology matrix.
Right.
And you definitely want to stick around, because later in this discussion, we're going to get into how this exact same technology allows a literal garage biohacker with a Wi-Fi connection and an API account to synthesize a pandemic-level pathogen.
Oh, man.
Bypassing every single physical biosafety lab on Earth.
Yeah, the sheer scale of the macroeconomics at play here is just staggering. Ultimately, I mean, this deep dive is about a massive wealth transfer.
Right.
Capital is rapidly fleeing the legally protected patent thickets of these legacy pharmaceutical conglomerates, and it is flowing directly into the physical infrastructure of digital biology.
Let's actually start right there with why that capital is fleeing in the first place. Because if we look back at, say the 1950s, drug discovery was relatively cheap.
Yeah, super cheap.
A small team of chemists could stumble onto something like penicillin or ibuprofen in a matter of months. It costs a fraction of what it does today. But now, the entire industry is ruled by something called Eroom's law.
Right. So Eroom is simply Moore spelled backward.
Okay.
Moore's law, as most people know, dictates that computing power gets exponentially cheaper and faster over time. Eroom's law is kind of the dark mirror of that concept.
Right.
Despite all our supercomputers, all our advanced robotics, our genomic sequencing, drug discovery is actually getting exponentially slower and more expensive every single year.
I always try to visualize this. It feels like the legacy pharma R&D model is basically taking a billion random keys, throwing them into a pitch black room, and just hoping one of them happens to fit into a lock that they can't even see.
That's a perfect analogy.
Because the low-hanging fruit, the easy drugs we could find by just testing natural compounds from tree bark or soil fungi, those have all been found already.
Exactly. And that structural inefficiency is why Big Pharma hasn't actually survived on scientific innovation for decades.
Wait, really?
No, they survive on legal monopolies. I mean, think about it. When a pharmaceutical giant gets incredibly lucky and actually finds a blockbuster drug in that dark room, they don't just patent the molecule itself.
Right.
They build a fortress around it. So they patent the method of delivery. They patent the specific dosage required. They'll patent the exact crystalline structure of the pill.
Oh, wow.
Yeah, they even patent the specific genetic biomarker of the patient it's intended for.
So even if a competitor manages to synthesize the exact same underlying chemical, they can't legally put it in a pill or prescribe it for that specific condition without triggering this massive lawsuit.
Right. It is a deliberate strategy to just stifle competition completely. And then right before that 20-year legal monopoly clock runs out, the exact moment generic competitors are gearing up to enter the market and drop the price of the drug.
Right, which is what's supposed to happen, right?
Right. Exactly what the system was designed for. But the conglomerate engages in a practice called evergreening.
Okay, wait, I want to dig into this, because if a patent is strictly capped at 20 years by law, how do they legally extend it? Are they just filing the exact same patent again?
No, they can't file the same patent. So what they do is they make microscopic, completely clinically irrelevant tweaks to the drug.
Really? Like what?
Like they might alter the binding agent inside the tablet, or they'll change the delivery mechanism from, say, a once-a-day pill to a once-a-week injection.
Oh, I see.
Or they might combine it with like a mild antacid and call it a brand new therapeutic.
Unbelievable.
Yeah, these aren't medical breakthroughs at all. They are aggressive legal maneuvers designed solely to reset that 20-year patent clock.
So they just lock it up again.
Right. It forces generic manufacturers to spend a decade and, hundreds of millions of dollars in court just to try and pierce that thicket. Yeah. I mean, the modern pharmaceutical industry is fundamentally a legal and financial enterprise that occasionally happens to sell medicine.
But that entire legal house of cards relies on one core assumption, doesn't it? It relies on the idea that discovering a brand new drug is impossibly hard.
Exactly.
Because if finding a new key in the darkroom takes 12 years and $2 billion, well, then holding the patent on your old key is worth its weight in gold.
Right.
So the question is, what happens to that business model when we finally turn on the lights?
The entire economic moat just vanishes overnight.
So let's talk about how the lights actually got turned on. Because to understand why this legacy model is collapsing, we have to look at the ultimate bottleneck of all human biology. And that is the protein. We aren't just single organisms. I mean, we are colonies of roughly 30 trillion cells.
Yeah.
And the actual workers inside those cells, the molecular machines that are firing your neurons, digesting your food, replicating your DNA, those are proteins.
Right. And the crucial thing to understand about a protein is that it starts its life as this long, one-dimensional chain of amino acids. There are 20 standard amino acids, and a typical protein might be a chain of hundreds of them, but it doesn't stay a flat string.
Right. It doesn't just float around like a loose piece of thread.
No, exactly. The moment that chain is created in your body, it folds, it twists, it bends, and binds into a highly specific, incredibly complex three-dimensional shape.
I always think of it like microscopic origami. It literally constructs itself into a physical tool.
That's a great way to put it. And that physical structure dictates absolutely everything. In biology, shape equals function. So if a protein folds correctly, it forms this perfect little pocket to, say, carry oxygen through your blood.
Right.
But if it misfolds, it clumps together in the brain and you get Alzheimer's or Parkinson's.
Oh, wow.
Or if the spike protein of a virus folds into the exact right shape, it acts as a physical key, unlocks your cell membrane, and infects you.
Therefore, if we want to cure a disease, we basically have to design a molecule that physically binds to a specific protein, either to alter its shape or to just block its function entirely.
Right.
Which brings up a major problem, because if we need to build a drug to bind to a protein, we need to know exactly what shape that protein is in the first place. And for 50 years, scientists were stuck trying to figure out how these strings of amino acids decide what shape they're going to take.
Yeah, they hit a massive wall. It's known as the Levinthal paradox.
Okay, break this down for us.
So back in 1969, a guy named Cyrus Levinthal did the math on protein folding.
Yeah.
And he calculated that a typical protein has so many possible ways it could fold into a 3D shape that if it just randomly tried every possible configuration to find the correct one, it would take longer than the age of the universe.
Just to clarify the school here.
Sure.
Longer than the age of the universe?
Yeah.
For one single microscopic protein to find its shape.
Yes.
That is insane.
It is. Yet inside your body right now, as we're talking, millions of proteins are folding into their correct functional shapes in milliseconds. It is a staggering computational feat performed by nature. And for decades, human scientists tried to predict how these proteins would fold using, brute force supercomputers and quantum chemistry.
They were just grinding through the math.
Right. Or they would spend years doing physical X-ray crystallography in these wet labs, literally freezing proteins and shooting X-rays at them just to map the 3D structure of one single protein.
Just one.
Yeah. We were completely flying blind.
So we were trying to design the keys without actually knowing the exact shape of the locks we were trying to pick.
Exactly.
But that brings us to the real case study that triggered this entire revolution we're seeing today. DeepMind and AlphaFold.
Yeah.
So DeepMind fed a neural network the known physical structures of about 100,000 proteins that scientists had painstakingly mapped over the last few decades.
Right. But the AI didn't just memorize those structures. By analyzing that massive data set, AlphaFold actually learned the underlying hidden rules of molecular physics. Yeah, it deduced the energetic forces that physically pull those amino acid chains together.
So it actually mapped the locks. Because AlphaFold predicted the 3D structure of almost every known protein on Earth. And it did it in a matter of days with near atomic accuracy.
Yeah, it was arguably the single greatest leap in the history of biology.
Right. But okay, if AlphaFold mapped every biological lock on Earth, that was an incredible achievement for understanding the human body. But if I have a patient dying of an aggressive tumor right now, understanding the lock isn't enough. I still need a new key.
Right.
So how do we jump from having a map to actually having a medicine?
Well, that is the pivot point where the industry is right now. Predicting the shape of a natural protein was only step one. The true gold rush began when AI moved from merely predicting biology to generating it.
Okay, this is where we get into the generative protein design.
Yeah.
Because we're also used to generative AI writing emails for us or, creating funny pictures of dogs in spacesuits or whatever. But there are AI models now, like RF diffusion, that operate on the exact same underlying mathematics. The same diffusion models that power those image generators. But instead of generating pixels, they are generating novel molecular machines.
Right. They are hallucinating enzymes. And it's important to understand these AI models do not search a database of existing drugs.
Right.
They mathematically generate entirely new molecular structures that haven't existed in 4 billion years of evolution.
Let's walk through how this actually looks in practice because it's mind-blowing. Say we're trying to cure a rare, really aggressive form of pancreatic cancer. The old legacy pharma way is to basically synthesize 100,000 random chemical compounds, physically dump them onto cancer cells in the petri dish, and pray that one of them is toxic to the tumor but somehow doesn't kill the patient.
Yeah, it's a brute force lottery, and it usually fails in human trials. But the generative biology model flips that entirely.
Right.
You start with a prompt, but the prompt is not text.
Right.
It's a set of physical constraints. You feed the AI the exact 3D topological map of a protein receptor that only exists on the surface of that specific pancreatic cancer cell.
So you essentially show the AI the lock on the cancer cell, and you give it parameters. You tell it, hey, design a novel, non-toxic protein binder that fits perfectly into this receptor. And when it binds, it must lock the receptor and trigger cellular apoptosis, meaning cell death.
Exactly.
Plus, it needs to be small enough to evade the human immune system, and it must remain stable at normal human body temperature.
Right. And you have to think about the computational scale of what you are asking that machine to do.
It's absurd.
It is. To fulfill that prompt, the AI has to search a latent space, which is essentially this multidimensional mathematical universe that maps every possible combination of amino acids.
Yeah.
And that space has roughly 10 to the power of 60 possible molecular configurations.
10 to the power of 60. I mean, that number is so astronomically large, it kind of breaks human comprehension. There aren't that many atoms in the Milky Way galaxy, are there?
No, not even close. And yet it searches that infinite space and generates millions of potential structures in seconds. Then it runs physics-based simulations on the fly. It digitally tests how strongly each of those hallucinated proteins binds to the cancer receptor. It filters out any designs that might accidentally bind to healthy heart tissue or liver cells. It optimizes the molecular weight for mass manufacturing.
And in 48 hours, literally 2 days, the AI takes that infinite universe of possibilities and hands you a short list of 10 perfect, bespoke molecular assassins.
Yeah.
10 novel proteins designed from scratch by a neural network perfectly tailored to kill that specific cancer.
Wow.
I mean, an army of human medicinal chemists working for a century couldn't compete with that velocity.
They absolutely couldn't. The cost of biological discovery just collapsed down to the cost of your AWS cloud compute bill.
Which changes everything. So we solved how to build a custom protein to attack a disease like cancer. But what if the disease isn't an external tumor or a virus? What if the problem is a typo in the patient's actual DNA blueprint? A custom protein can't rewrite genetic code, can it?
It can if it's designed to interact directly with the genome, and this is the transition to CRISPR 2.0. We are moving from using molecular scissors to using a molecular word processor.
Okay, because the first generation of CRISPR-Cas9, was highly publicized. I mean, everyone heard about it. You programmed a guide RNA to find a specific sequence of DNA, and the Cas9 enzyme acted as a literal blade, right?
Exactly.
Just cutting of the double helix at that spot to disable a bad gene.
Right. But the problem with that sledgehammer approach is what happens next. When you induce a double-strand break in human DNA, the cell recognizes it as catastrophic damage.
She freaks out.
Yeah, it panics. It uses an emergency repair mechanism called non-homologous end joining.
I've heard this described as molecular duct tape. Like the cell just hastily jams the broken ends back together so it can survive.
Yeah, and it's incredibly messy. That panic repair is prone to massive unintended chromosomal deletions. Those off-target mutations can completely scramble healthy genes and ironically actually trigger cancer.
Oh, man.
Yeah, first-gen CRISPR was amazing for lab research in petri dishes, but it proved highly dangerous and unpredictable for actual human therapeutics.
But now we are looking at base editing and prime editing. And I like to think of base editing as a molecular pencil rather than a pair of scissors. How does this system actually fix a typo in the DNA without cutting the strand?
So it uses a modified version of the Cas9 protein that has been deactivated. They call it a dead Cas9.
Okay.
It can no longer cut DNA. Instead, it acts purely as a GPS unit. It navigates through the genome, finds the exact genetic typo, and just parks there.
Right.
And attached to this GPS is a completely different chemical enzyme. So if a patient has a genetic disease caused by just one single incorrect letter, say they have a C where there should be a T,
Yeah.
the base editor reaches in, chemically converts the C into a T, and leaves the entire DNA spine completely unbroken.
No double strand break, no cellular panic, no molecular duct tape, just an elegant, localized chemical rewrite.
Yeah, breathtaking precision. And prime editing takes this even further. It is a literal search and replace function for the human genome. It can delete whole sentences of DNA, insert new paragraphs of genetic code, and rewrite complex genetic circuits with near zero off-target effects.
Wow. But the obvious hurdle here is delivery, right?
Yeah.
If I have a patient with a neurological disease, how do I actually get this molecular word processor into their brain? I can't just inject it into their arm and hope it finds its way.
Right, delivery has always been the bottleneck, but now we use AI-designed lipid nanoparticles, or LNPs.
Oh, like the ones used in the mRNA vaccines.
Exactly. These are microscopic spheres of fat that encapsulate the editing machinery. And we can actually prompt our AI models to design the outer surface of these fat bubbles so they exhibit specific targeting behaviors.
That's crazy.
Yeah, we can program them to bypass the liver, cross the highly restrictive blood-brain barrier, and fuse directly with the patient's motor neurons. The LNP delivers the prime editor into the cell, the code gets rewritten, and the disease is permanently cured at the source code level.
So when you combine generative AI, hallucinating the exact molecular machinery you need, and CRISPR 2.0 safely installing it into the genome, you completely dismantle the legacy pharma business model.
Entirely. The underlying science dictates the shift in capital.
Okay, so if I'm listening to this deep dive, and I'm realizing that the entire global healthcare infrastructure is essentially being rebuilt from the ground up, I need to know how to position myself.
Right.
I want to shift into the playbook. What are the specific steps an investor or professional can research this week to capture the value of this massive transition?
Yeah, there are 4 highly actionable steps you can investigate right now to map out where the capital is flowing.
Okay, let's hear them.
Step 1 is screening for the TSMC of biology.
Okay, so just to explain that reference, in the digital computing world, NVIDIA designs the brilliant AI chips, but they don't actually build them. They contract TSMC in Taiwan to physically print the silicon. So TSMC basically sits as the ultimate toll booth for the entire digital economy.
Exactly. And we are seeing the exact same dynamic play out in synthetic biology. The AI model might design a brilliant sequence of genetic code, but that code is just digital data on a screen.
It's just a file.
Right. You still have to physically synthesize it into actual strands of DNA to test it in a lab. So there are mid-cap companies operating massive high-throughput DNA foundries right now. They hold the core patents on high-fidelity enzymatic DNA synthesis.
They are the physical printers.
Yes, turning digital A, C, T, and G code into physical biological material.
Yeah.
Every AI startup, every university, every cloud lab has to pay them. They are the toll booths of the biorevolution.
Okay, step 2 in the playbook. Investigate cloud lab picks and shovels.
Right.
We've talked extensively about how fast the AI moves, but testing those AI designs in the real world requires incredibly specialized physical hardware.
It does. You should be looking for industrial automation firms, specifically the companies that are experiencing multi-year backlogs for things like automated liquid handlers, robotic cellular incubators, and high-throughput mass spectrometers.
Just to ground that term real quick, a mass spectrometer is essentially a highly complex machine that weighs molecules to figure out exactly what they're made of, right? You use it to verify that the protein you tried to build actually matches the digital design.
Correct. And these automation firms are essentially selling the physical infrastructure of the AI revolution.
Yeah.
They don't take on the biological risk. They don't care which biotech startup actually cures cancer. They get paid every time a robotic arm moves a test tube or a spectrometer runs a sample. So look for firms that have explicitly adapted their hardware for these new CRISPR workflows.
That makes total sense. Now, step 3, identify short targets in legacy pharma. Because if the era of the blockbuster drug is dying, someone is going to be left holding the bag.
Absolutely. You want to research publicly traded conglomerates with massive bloated R&D divisions. Look for companies with high revenue exposure to expiring patent cliffs, those evergreen portfolios we discussed earlier. And most importantly, look for low internal AI adoption.
Right, the ones fighting the trend.
Yeah. Historically, these giants would just buy early-stage biotech startups to replenish their pipelines. But the premium they use to pay for those acquisitions is going to plummet because the fundamental discovery process is being commoditized by software. Betting against the companies that refuse to adapt is a highly asymmetric macro trade right now.
Wow, okay. And finally, step 4, track in silico clinical trials. Because we've solved discovery, we've solved synthesis.
Okay.
But the biggest bottleneck left in all of this is the FDA.
Yeah, human clinical trials take years and cost billions.
So how does software possibly bypass a federal mandate for human testing?
By mathematically proving safety before a drug ever touches a human being. There are startups building what they call digital twins of human organ systems.
Okay.
They train deep learning models on massive data sets of multi-omic data.
Multi-omic, meaning they aren't just looking at your DNA, they are looking at genomics, proteomics, metabolomics, all the intersecting layers of biological data combined.
Exactly, it's the full picture.
Yeah.
And using that data, you can simulate exactly how a novel AI-designed drug will metabolize in a digital liver, or how it will interact with a digital cardiovascular system.
Oh wow, so you skip the guinea pig phase entirely.
Right. The companies that can mathematically prove a drug's safety in silico, drastically reducing the failure rate and the need for massive human phase 1 trials, they're going to become the new regulatory gatekeepers of the global healthcare system.
That is wild. But we need to take a step back here.
Yeah.
We've talked about the medical miracles, we've mapped out the wealth playbook, but we have to talk about the dark side. The garage biohacker.
Yeah, this is the scary part.
Because when you democratize the ability to write the code of life, you inherently democratize the ability to write the code of death.
It's the classic dual-use dilemma. The exact same technology that cures cancer can be inverted to cause mass devastation.
And historically, if you wanted to create a novel biological weapon, the barrier to entry was massively physical and financial. I mean, you needed a state-sponsored budget. You needed a biosafety level 4 lab with physical airlocks, specialized hazmat suits, and a whole team of rogue virologists.
Right.
Are you saying someone can bypass all of that physical infrastructure today?
The physical barrier to entry has completely collapsed. It is now purely informational. The genetic sequences for almost every known virus on Earth, including highly lethal strains of influenza, Ebola, and coronaviruses, they are just sitting in open source databases online. Anyone can download them.
Anyone. Walk me through the scenario that is literally keeping national security agencies awake at night right now. The amateur hacker operating out of a rented garage.
Okay, imagine an individual with just a Wi-Fi connection, a Cloud Lab API account, and a secondhand PCR machine they bought online.
Just to jump in, a PCR machine polymerase chain reaction. It's essentially a DNA photocopier that allows you to multiply small segments of DNA, right?
Right, and they're relatively cheap now. So they take the exact same open source generative AI model we just discussed for curing pancreatic cancer, but they alter the prompt.
Oh no.
They ask the AI to design a novel protein spike that actively evades the human immune system. Or they prompt it to increase the binding affinity of, say, an avian flu virus to human lung tissue.
So they write the malicious prompt. The AI hallucinates the bioweapon right there on their laptop.
Yes, and then they send the Python script via API to an automated cloud lab in another country. The robotic facility synthesizes the lethal DNA, packs it in a vial, and mails it back to the garage via standard shipping.
Right, they just bypassed every single physical BSL-4 biosafety protocol on the planet without ever putting on a pair of gloves.
Exactly, which means the ultimate national security threat is no longer a physical border, a standing army, or even scanning for nuclear materials. The front line of defense is now the cybersecurity of biological data.
Wow.
It relies entirely on the KYC, the Know Your Customer screening protocols utilized by these DNA synthesis foundries.
Right, because if they don't catch it,
Right. If a foundry's algorithm fails to flag a malicious sequence that's hidden inside a benign order, the weapon gets printed. We are literally handing the power of planetary evolution to anyone with an internet connection and a credit card.
that is just chilling. And if you're listening to this, you might be thinking this sounds like a science fiction movie. But there is a second dark side here that is far more insidious than terrorism, and it's happening right in plain sight.
Yeah.
We are staring down the barrel of a permanent genetic class divide. I call it the Elysium scenario.
That's exactly what it is.
Because you might look at this tech and assume that as it scales, these gene therapies will become cheap for everyone, just like flat screen TVs or smartphones. But that's not how bespoke medicine works, is it?
No, it's going to create a severe economic disparity because the crucial difference here is that these are N of 1 therapies.
Right.
They are custom designed for a single individual's exact genome. You cannot mass manufacture them on an assembly line. And because of that bespoke nature, the initial costs of these AI-designed gene therapies are going to be astronomically high.
So we aren't talking about equal access to healthcare.
Not at all.
We're looking at a future where the ultra-wealthy have the capital to literally purchase better, longer, optimized genomes for their children.
Yeah.
They will pay to edit out the genetic markers for Alzheimer's and heart disease. They will pay to enhance muscle density, optimize metabolic efficiency, and extend cellular lifespan.
While the working class will be left relying on the traditional reactive chemical-based healthcare system we have today, they will be stuck managing chronic decline with legacy pills, while the elite are proactively rewriting their source code for longevity.
It fundamentally breaks the concept of human equality.
It does. It raises profound questions about the very definition of humanity itself. This isn't just a wealth gap, it is a biological speciation event. You will eventually have two classes of humans functioning on fundamentally different biological hardware.
And you know there will be a massive, visceral, societal reaction to that. I mean, people aren't just going to quietly accept that the rich get to become superhumans while everyone else just decays.
No, it's going to trigger fierce, deeply emotional political movements. We will see the rise of bioluddites.
Bioluddites, yeah.
They will demand draconian global bans on human germline editing. Those are the edits that can actually be passed down to future generations.
Right, because once it's in the germ line, it's permanent for that family line.
Exactly. They will frame synthetic biology not as a medical miracle, but as this unnatural dystopian violation of the human condition. The regulatory war over who gets to edit the human source code and, frankly, who owns the copyright to those edits is going to be the most vicious legal battle of the 21st century.
It's going to be chaotic. But I want to pivot here and actually challenge a narrative we hear constantly in Silicon Valley right now. Because every tech founder and venture capitalist keeps repeating the same mantra that software is eating biology.
Right.
They act like the AI algorithm itself is the ultimate impenetrable moat. And I think they were entirely wrong.
I agree. The AI algorithm is absolutely not the moat.
Because open source models are going to inevitably commoditize generative biology software.
Yeah.
I mean, if a massive tech company spends $1 billion to build the ultimate protein folding model today, a decentralized group of researchers is just going to release an open source version that is 90% as good next year.
Always happens.
Right. So if the software is that accessible, why wouldn't I just dump all my investment capital into the startup with the smartest AI prompt engineers?
Well, because that assumption leads directly to a massive trap. And it's a trap that has bankrupted numerous naive tech founders and investors recently.
The investor regret is very real here. I mean, you have smart people from the software world pouring millions of dollars into pure play biological AI startups, believing that a clever AI prompt alone cures a disease.
Right.
They see the AI hallucinate a beautiful, perfectly folded 3D structure on a computer screen. The digital simulation says it works, and they think the job is done.
Right. But the reality is, Biology is not clean code.
No.
You cannot compile a human cell like you compile a software app. You have to close the loop with messy, chaotic, physical biology.
You have to prove it actually works.
Exactly. You have to prove that the hallucinated protein doesn't just fold correctly on a screen, but that it actually folds correctly in a real physical petri dish without turning into, a useless clump of toxic sludge.
Which is why the true trillion-dollar mode is the physical infrastructure.
Yeah.
The cloud lab. Biology as a service. I want people to imagine a warehouse the size of a football field, completely pitch black inside, completely devoid of humans. Thousands of robotic arms, automated liquid handlers, and mass spectrometers running millions of physical wet lab experiments 24 hours a day, 7 days a week.
Right. And the process looks like this. The AI hallucinates a novel protein structure. It sends Python code via API to this dark warehouse.
Okay.
The robotic foundries physically print the DNA. The liquid handlers synthesize the protein. They drop it onto living human cells in an incubator. They run the mass spectrometry to analyze the results. And then, and this is the critical part, they feed that pristine physical outcome data back into the AI model.
Oh, so the AI learns from the physical friction of the real world?
Exactly. The data set generated by those physical experiments is the actual moat, not the digital algorithm.
Yeah.
The companies generating the real-world friction data are the ones who will control the industry.
That makes perfect sense. So when we project this entire ecosystem forward, the 10-year outlook for the industry is essentially a complete restructuring of intellectual property.
It is.
I think we're looking at the absolute death of the biological patent.
Yeah, I agree.
But how does a patent actually die if it's a legally binding document? Big Pharma has literal armies of lawyers defending these thickets.
Because the legal framework of a 20-year monopoly is totally incompatible with an artificial intelligence that iterates in days.
Oh.
We are witnessing the rise of a GitHub for DNA. Open source biological repositories are just exploding globally. Researchers everywhere are uploading the genetic sequences of novel enzymes, CRISPR guide RNAs, and synthetic pathways.
Okay, so think about what that means for a legacy conglomerate. Say they hold a lucrative patent on a specific biological pathway that generates $1 billion a year. A startup with an open source AI agent doesn't need to fight them in court for 10 years.
No, they don't.
They simply prompt the AI to invent around the patent.
Exactly. They instruct the AI, hey, analyze this patented protein. Now hallucinate a completely different, structurally distinct protein that achieves the exact same biological function, but is chemically different enough that it does not infringe on the existing patent.
And the AI designs a functional workaround in 48 hours. The cloud lab synthesizes it, tests it, and uploads the verified sequence to the open source matrix. A multi-billion-dollar, 20-year legal monopoly is rendered economically worthless in a matter of weeks.
Yep. You cannot litigate against an algorithm that can invent a million viable workarounds before the judge even bangs the gavel in the first lawsuit.
Which means the era of the blockbuster drug, one mass-produced molecule meant for millions of people protected by an impenetrable patent, is completely over.
It's done. It is being replaced entirely by the N of one therapeutic.
Custom drugs. Designed, synthesized, and manufactured for a single specific patient based on the unique blueprint of their genome.
It is a profound shift in what it means to be human.
Let's bring this home with a 3-bullet recap of everything we've mapped out in this deep dive.
Sounds good.
First, the legacy pharma model, reliant on Eroom's law and dense legal patent thickets, is mathematically broken and is actively being incinerated by generative AI.
Yep.
Second, biology is no longer a science of blind discovery. It is an engineering and cloud discipline. The real wealth is moving into the physical infrastructure, the DNA synthesis foundries acting as the TSMCs of biology, and the robotic cloud labs generating physical friction data.
Exactly.
And third, the democratization of this technology creates massive existential risks, ranging from Wi-Fi-enabled bioterrorism executed in a garage to a permanent, irreversible genetic class divide.
Yeah, the underlying code of life has been cracked, and we are just beginning to write our own scripts.
Which leads me to a polarizing question for everyone listening. I really want you to think about the reality of this technology. If an open source AI could design a custom life-saving gene therapy for a terminal disease, and it could synthesize it for $50 in 4 hours, but that therapy has not passed a 10-year FDA clinical trial, would you inject it to save a loved one? Do you trust the algorithm more than the regulatory state? Drop your answers in the comments.
That is the ultimate ethical debate that will define the next century of our evolution.
If this deep dive gave you a terrifying new perspective on the microscopic machines inside your veins, hit subscribe to MediaGenX. Next week, we're zooming out from the microscopic to the planetary to discuss the AI climate cartel and how tech monopolies are literally patenting the weather.
Stay curious, stay positioned.
And remember, not financial advice, do your own research. But seriously, 12 years and $2.5 billion for a key that might not even turn. The lights are finally on, the locks are mapped, the matrix is open.
See you next time.